Hello students, welcome to Read Med Prep Academic channel. Today we are going to discuss in sexual reproduction in plants part 5 about fertilization in plants. The fusion of the male and the female gamete is called fertilization. The fusion of one male gamete with the female gamete and another male gamete with the secondary nucleus or the polar nuclei is called double fertilization. It is seen in angiosperm. Fusion of the one male gamete with the secondary nucleus, that is the two polar nuclei present in the ovule, is called triple fusion. Since there is involvement of one male nucleus and two polar nuclei, it is called triple fusion. Here you can see in this image, the pollen grains fall on the stigma, and the pollen grains form the pollen tube and enter through the micropyle into the ovule, and the one male gamete fuses with the egg nucleus, and this fusion is called syngamy. And the fusion of the another male gamete with the secondary nucleus containing the two polar nuclei is called triple fusion. What are the events of fertilization? Pollen falls on the stigma, pollen tube grows in the style, entry of pollen tube in the ovule, entry of pollen tube in the embryo sac, double fertilization and triple fusion. What are the stages in double fertilization? Pollen falls on the stigma, germination of the pollen to form the pollen tube, growth of the pollen tube in the style, direction of the pollen tube towards the micropyle of the ovule, entry of the pollen tube into one of the synergids of the embryo sac, discharge of the male gametes, and syngamy and triple fusion. Now let us discuss one by one. When the pollen falls on the stigma, in nature, a variety of pollens fall on the receptive stigma, but all of them do not germinate and bring about fertilization. The receptive surface of the stigma receives the pollen. If the pollen is compatible with the stigma, it germinates to form a tube. This is facilitated by the stigmatic fluid that is present, and this type of stigma is called wet stigma. And sometimes it is facilitated by the pellicle. This type of stigma is called the dry stigma. These two, the wet and the dry stigma, decide the incompatibility and compatibility of the pollen through the recognition-rejection protein reaction between the pollen and the stigma surface. Sexual incompatibility may exist between different species. That is called interspecific incompatibility, or sexual incompatibility between the members of the same species. This is called intraspecific incompatibility. This intraspecific incompatibility is also called self-incompatibility. Here in this image you can see that the pollen grain belonging to S1 and S2 plants falls on the stigma. It is incompatible with the stigma of S1 S2 type and S1 S3 type, whereas the same pollen grain of the plant S1 S2 when falls on the stigma and the pollen tube develops in the style of S3 and S4 plant. So this compatibility is also very important in fertilization. The first visible change in the pollen soon after it lands on the stigma is hydration. The pollen wall proteins are released from the surface. Here you can see in this image the pollen stigma interaction. The pollen first falls on the sticky stigma. This pollen grain adheres to the stigma surface, and there is absorption of water by the pollen grain and the swelling of the pollen grain. And when the pollen grain is compatible with the stigma, the germination of the pollen tube occurs and the pollen tube penetrates the stigma wall and enters into the style. And the pollen tube grows into the style and reaches the micropyle of the ovule. During germination, the entire content of the pollen moves into the pollen tube. Here on the right side you can see the contents into the pollen tube having the cytoplasm along with the organelles and the tube nucleus and the two male gametes. The growth is restricted to the tip of the tube and all cytoplasmic contents move to the tip region. The remaining part of the pollen tube is occupied by a vacuole which is cut off from the tip by the callose plug. Here in this image you can see that the pollen tube contains all the components of the cytoplasm, including the Golgi apparatus, the endoplasmic reticulum, the mitochondria, the microfilaments. Along with it, there is a vegetative nucleus and the two male sperm cells. When the contents move into the pollen tube, there is a vacuole formed, and this cuts off the contents from the main pollen grain body by forming a Picado's plug. The extreme tip of the pollen tube appears hemispherical and is transparent when viewed through the microscope. This is called cap block. As soon as the cap block disappears, the growth of the pollen tube stops. After the germination of the pollen tube enters into the style from the stigma. The growth of the pollen tube in the style depends upon the type of the style. There are three types of styles: a hollow style, which is otherwise called the open style, a solid style, otherwise called the closed style, and a semi-solid style, otherwise called the half-closed style. A hollow style, when you take, it is common among the monocots. It has a hollow canal running from the stigma to the base of the style, and the canal is lined by a single layer of glandular cells. This tissue is called the transmitting tissue. This transmitting tissue secretes mucilaginous substances. The pollen tube grows on the surface of the cells lining the stylar canal. The canal is filled with secretions, which serve as the nutrition for the growing pollen tubes and also controlling the incompatibility reaction between the style and the pollen tube. The secretions contain carbohydrates, lipids, and some enzymes like esterases, acid phosphatases, as well as compatibility controlling proteins. Now, when you go to the closed type or the solid style, it is most common among dicots. It is characterized by the presence of central core of elongated, highly specialized cells. These cells are called transmitting tissue. This is equivalent to the lining cells of the hollow style and does the same function of nutrition to the growing pollen tube. Its contents are also similar to the content of those cells. The pollen tube grows through the intercellular spaces of the transmitting tissue. Now, when you move on to semi-solid style or the half-closed style, this is the intermediate between the solid and the open type. There is a difference of opinion on the nature of the transmitting tissue. Some authors consider that it is found only in a solid styles, while others consider the lining cells of the hollow style also have transmitting tissue. Now, let us move on to the entry of the pollen tube into the ovule. There are three types of pollen tube entry into the ovule. One is porogamy, number two is chalazogamy, and number three is mesogamy. Now, what is porogamy? When the pollen tube enters the ovule through the micropyle, it is called porogamy. You can see in the image on the right side. What is chalazogamy? When the pollen tube enters the ovule through the chalaza, this is called chalazogamy. You can see in the image on the right side. What is mesogamy? When the pollen tube enters the ovule through any part of the integument, this is called mesogamy. You can see in the image on the right side. Now, entry of the pollen tube into the embryo sac. Irrespective of the entry of the pollen tube, whether it is porogamy or chalazogamy or mesogamy, the pollen tube enters the embryo sac only at the micropylar end. The pollen enters into the embryo sac directly into one of the synergids. Here you can see in this image on the left side the three antipodal cells, the polar nuclei, which are two in number, the egg nucleus, and the two synergids, and the pollen tube coming close to the ovary. And when it enters through the micropyle, it enters through one of the synergids. In this image on the right side, you can see the pollen tube containing the tube nucleus and the sperm nuclei. The growth of the pollen tube towards the ovary, ovule, and the embryo sac is due to the presence of chemotropic substances. Now, what is obturator? The pollen tube, after traveling the whole length of the style, enters the ovary locule, and this is guided towards the micropyle of the ovule by a structure called obturator. You can see in the image on the right side the obturator tissue which guides the pollen tube towards the micropyle. After reaching the embryo sac, a pore is formed in the pollen tube wall at its apex or just behind the apex. and the contents of the pollen tube, the two male gametes, the vegetative nucleus or the tube nucleus and the cytoplasm are discharged into the synergids into which the pollen tube enters. Here you can see in the image on the right side, the tube enters through the filiform apparatus into one of the synergids and the contents are discharged into one of the synergids. The pollen tube does not grow beyond this level once it enters the synergids. In the embryo sac, the tube nucleus that is present disorganizes. S.G. Navashin and L. Guingard in 1898 and 1899 observed in Lilium and Fritillaria that both the male gametes released from a male gametophyte are involved in the fertilization. They fertilize the two different components of the embryo sac. Since both the male gametes are involved in the fertilization, the phenomenon is called double fertilization. And this double fertilization phenomenon is very unique in angiosperms. In this image, you can see that the one male gamete fuses with the egg nucleus and forms the zygote, which contains 2n number of chromosomes. And another male gamete or the sperm fuses with the secondary cell containing two polar nuclei, forming the 3n cell, which in future forms the endosperm. One of the male gametes that fuses with the egg nucleus forms the zygote. This fusion is called syngamy. The second gamete migrates to the central cell where it fuses with the polar nuclei or the fusion product, the secondary nucleus, and forms the primary endosperm nucleus. Here in this image you can see on the left side is the pollen grain, growing pollen tube having the vegetative tube nucleus and two male nuclei. On the right side you can see the ovule containing the antipodal cells having the n number of chromosomes, the secondary nucleus which is formed by the two polar nuclei having 2n chromosomes, and the egg which has n number of chromosomes. One male nuclei fuses with the egg nucleus. So when 2n nuclei are fused, 2n zygote is formed. Another male nuclei fuses with the secondary nucleus which has 2n number of chromosomes, and this forms a 3n endosperm. The zygote forms the embryo which is embedded in the seed, and the endosperm nuclei forms the endosperm. The endosperm and the embryo together form the seed. Since this involves the fusion of three nuclei, that is one male nuclei and two polar nuclei, this phenomenon is called triple fusion. This triple fusion cell results in the primary endosperm nuclei. This forms the endosperm, which gives nutritive tissue of the embryo, or which forms the nutritive tissue of the embryo. Here in this image you can see the fusion of one male gamete with the egg nucleus forming the zygote, and another male nuclei fusing with the secondary nucleus forming the endosperm, and the fusion of both the male nuclei with two different components is called double fertilization. In this image you can see on the left upper side a male nucleus fuses with the egg cell nuclei to form the zygote, and another male gamete fuses with the secondary nuclei and forms the 3n endosperm. The fusion of two nuclei with the two different components of the ovule is called double fertilization. The fusion of one male gamete along with the secondary cell having two polar nuclei is called the triple fusion. The triple fusion results in the formation of the endosperm, and the syngamy results in the formation of zygote, which helps in the formation of the embryo. So today In sexual reproduction in plants part 5, we have discussed about fertilization in plants. Thank you. Kindly subscribe, like, share and comment the channel Read Med Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.